welcome to the video on mitigation strategies for nitrosamine drug substance related impurities. These are the nitrosamine impurities having the structure similar to the API or drug substance. That's why these impurities are called as nitrosamine drug substance related impurities and abbreviated as NDSRIs. So let's start with the video for the mitigation strategy for NDSRIs. Risk factors for nitrosamine formation are amine systems. So model amine systems used to study nitrosamine formation in the oral formulations. Key risk factors identified are as pK values for the amines, the addition of water during processing like wet granulation, form of amine like salt or free base, physical stability of amines, crystalline versus amorphous. These are the key risk factors for the NDSRI's formation. pK values, water addition or use of water like process in the wet granulation, or presence of the free amine versus salt or free base amines, and physical stability of amines related to the API polymorphic form, whether it is crystalline or amorphous. Reducing the nitrosamine levels without scavengers is the way for reducing nitrosamine and NDSRIs. So key findings are the nitrate levels, not amine levels, are the rate limiting factors in the NDSRI formation. So emphasis is more given on the nitrate levels. Effective mitigation strategies involves select excipients with low nitrate levels to reduce nitrosamine levels by 10x. If you use the excipients with nitrate levels from 0.04 to 0.1 ppm, then you can reduce the nitrosamine and NDSRI to a great extent. For high risk formulation, additional strategies like wet granulation to direct compression transition or incorporating nitrate scavengers may be necessary. So if your formulation is high risk, then you can change the manufacturing method from wet granulation to direct compression so that you can avoid the use of water and use of heat during drying process. Also, you can incorporate nitrate scavenger or nitrate scavenging agents to reduce the NDSRI formation. So goal is to reduce the nitrosamine levels and NDSRI levels to the acceptable limits or acceptable daily levels. Control strategies for impurity amines in the APIs. Key considerations are impurity amines, spatial location in the API influences nitration kinetics or nitrosation kinetics. Crystallization, crucial in rejecting and controlling impurities. Sleep test, SLIP testing is there. SLIP stands for solubility limited impurity purge. Test to identify when impurity forms a pure solid phase during crystallization. Coming to the effectiveness of antioxidants and nitrate scavenging agents. Antioxidants can be used like ascorbic acid is there, caffeic acid is there, ferulic acid is there, and other antioxidants are also there which decrease the nitrosamine and NDSRI's formation. So other strategies includes increase antioxidant concentration for better NDSRI mitigation, maintaining a neutral pH as protective strategy. So you can use the basifying materials and bring down the acidic pH to neutral pH or little bit basic pH. Impact of manufacturing conditions. Elevated heat and moisture during wet granulation can promote NDSRI formation. Direct compression in continuous manufacturing may aid in mitigation. So if your products are prone to nitrosamine and NDSRIs and are being manufactured by the wet granulation followed by drying using heat, then you can change the method to the direct compression. Nitrate scavengers in NDSRI mitigation. Nitrate sources are present in the water, APIs, excipients, and atmospheric nitrogen sources. Then you can use effective scavengers like PABA, para-aminobenzoic acid, and it is identified an efficient inhibitor of N-nitrosamine formation, comparable efficacy to ascorbic acid and L-cysteine. Considerations for selection. So consider compatibility with the amine structure, API, excipient and the dosage form whenever you are going to select the nitrite scavengers. Regulatory considerations for NDSRI mitigation. Mitigation strategies involves FDA encourages to explore various strategies to reduce NDSRIs to acceptable levels. So FDA and other regulatory bodies encourage the exploration of or exploring the various strategies to reduce NDSRI formation. Submissions may require bioequivalence bridging studies for reformulated drugs. So if you have changed manufacturing process or you have incorporated some excipient to bring down the NDSRI at acceptable level, then there may be a chance that bioequivalence testing is required. Review process. FDA may request meeting packages for companies proposing innovative mitigation strategies. Ongoing dialogue with the FDA encouraged to ensure compliance and safety. So this is regarding the FDA. They can aid the companies for promoting the innovation in the reduction of nitrosamines. Additional mitigation strategies and analytical methods. Further topics discussed are like advanced analytical methods for detecting NDSRIs, other mitigation strategies for different drug forms and APIs. So mitigation strategy is not the same for all the molecules and formulations, but it may be different for each of the different category. Mitigation strategies should be tailored to specific formulation, APIs, and manufacturing processes. Detailed data, including stability studies and impurity profiles, may be required for the approval. So, this is regarding the regulatory considerations. Detailed data is required, stability studies are required, impurity profiling is required for the approval. This is a brief overview for the mitigation strategies for nitrosamine drug substance related impurities. Many videos are available onto this channel for nitrosamines and NDSRIs. You can visit those videos and can have a good idea for these topics. Thank you for watching the video and please do like, share and subscribe to this channel.